Okay, welcome back. So we're going to transition now. We're going to go from the world of DNA to the RNA world. And we're going to start with prokaryotes. And the reason we start with prokaryotes is a much simpler story. So the principles that I'm going to describe will apply to eukaryotes, but there'll be some important differences. Okay? So the objectives, what we want to try to do is introduce you to the various processes that bacteria use to make RNA, and specifically messenger RNA. Because remember, there's multiple types of RNA. Messenger RNA, ribosomal RNA, transfer RNA, the various small non-coding RNAs, some large non-coding RNAs. There's a whole bunch of really cool RNA molecules. But today we're going to focus on how does a bacterium make messenger RNA, and in subsequent lectures we're going to talk about how do bacteria and mammalian cells make protein from messenger RNA. And again, we're going to try to spin this from sort of the medical applied perspective. And the last point on the objectives list kind of makes that point is I want you to understand how this drug, rifampin, is used to treat tuberculosis. So let's start there. So the central dogma of molecular biology is illustrated here, and it's different in eukaryotes and us versus prokaryotes. So if you don't know this pathway, if you don't have this sort of in your head, study this and learn these various names and steps, because we're going to refer to this over and over again as we go through these lectures. Now, the prokaryotic side is pretty simple, right? The gene structure is pretty simple, no introns. It makes messenger RNAs, which often code for multiple proteins, what are called operons. And then translation produces proteins, and all of this kind of goes on along with DNA replication and DNA repair pretty much at the same time in the same compartment, right? Just in the cell. These cells don't have true nuclei. They kind of have clusters of structures where this stuff occurs, but not organized into true nuclei like our cells are. So those are some profound differences in the molecular biology, which then translate to profound differences in the sensitivity of these processes to certain drugs, to chemical inhibitors, some of which are useful in the clinic. And one of those drugs is a molecule called rifampin. So what's this about? So our story here is about tuberculosis, a lung disease, a lung infection. So the subject here is a 36-year-old woman admitted to the hospital. So a history of cough, which became productive. Okay, so there's your first clue: cough. Why does she have a cough? She's got fatigue, night sweats, and weight loss. Again, these are probably important signs that you should wonder, why is she fatigued? Why is she sweating? And what's the deal with the weight loss? Now, she's diabetic. That's a comorbidity that's a confounding factor in any infection. And she has poor nutritional status. Again, a confounding factor in dealing with infections. She's a volunteer at a nursing home, and 2 of her patients were diagnosed with active tuberculosis. So she's got exposure. All right? So this is a way that when you read these vignettes, you need to dissect what's important signs, what are the important symptoms that she reports, and what's the important history. So the key history here is obviously she's a healthcare worker, so she's exposed to all manner of interesting things, including TB. All right, so how about some lab results? Chest X-ray revealed bibasilar infiltrates. More about that in just a sec. She had a positive PPD test, so the skin prick test, put some tuberculosis antigen on the skin and see if there's antibody reactivity, and the answer was yes. And then that alone is pretty indicative of she's either infected or had been infected in the past. And now, is the infection active? Take a sputum smear and a culture, and what they find is acid-fast bacilli. Okay? And again, that's a diagnostic test that would suggest tuberculosis. HIV test was negative. So here's the lung X-ray. Okay. So the bibasilar infiltrates. Basically what that means is you get fluid in the alveoli. The alveoli don't contain fluid normally. They contain air. Right? Well, air is a fluid. They don't contain water, put it that way. All right, so they don't show up as little white dots on an X-ray. And here you see little white dots in the basilar surface. Okay, so there's something wrong. She's got an infection. So how would a physician treat this infection? And the answer is complicated. Tuberculosis responds to a variety of antibiotics and antimicrobial drugs, but now tuberculosis has actually become a much more serious disease than it was even 10, 15, 20 years ago, 30 years ago when I started teaching this stuff. Drug-resistant tuberculosis has become a huge clinical problem. So typically, it's treated with a combination of drugs. Rule of thumb in most infectious disease is figure out what the infecting agent is, use the single most effective, least toxic, and reasonably cost-effective treatment. Tuberculosis is the other way around. Always use multiple drugs. And that's because the pathogen grows slowly and resistance develops to each of the drugs. So you combine drugs to try to essentially defeat the resistance. More on that in a second. So these are the names of the drugs: isoniazid, ethambutol, pyrazinamide, and rifampin. So we don't care right now what these other agents do, but we want to know, well, what does rifampin do, and why is this interesting in terms of RNA synthesis? Well, what rifampin does is it has a fairly selective action on the RNA polymerase. That's a new idea, right? An RNA polymerase that bacteria make, most bacteria. So here's rifampin. It's kind of this funny structure down, ring structure. What it does is it binds to a particular subunit of the RNA polymerase, and in this application, the mycobacterial polymerase, which, by the way, it's sort of represented as a big blue machine, but this is really a multi-protein complex. There's lots of different subunits. So rifampin binds to one subunit, which happens to manage the exit of the newly synthesized RNA from the exit channel. Okay, so it binds right there. And so it inhibits the RNA polymerase. It blocks messenger RNA synthesis, which will slow down or inhibit the growth of the mycobacterium. And when combined with those other drugs, you generally get a pretty good response. It takes a long time, 3 months, 6 months, 12 months, to eradicate the disease, but it can be done. Now, the problem with rifampin is it's pretty good when combined with those other drugs for tuberculosis, but when used all by itself as an antibiotic, it's actually pretty crappy. And the reason is that resistance occurs pretty frequently. So it's rarely used as a single-dose therapy against other bacteria. So knowing where it works, okay, binding to, sorry, keep flipping around, excuse me, back, okay, thank you. Knowing that it binds to a particular subunit of this polymerase, how could resistance arise? What's a likely possibility? What do you think? Sort of think mechanistically, think molecular biology, think about genes. What could happen to make that enzyme resistant? Sorry? Yeah, absolutely right. So the subunit it binds to, that gene is susceptible to a fairly high mutation rate. And when the drug's used all by itself, bacteria almost always become resistant because of mutation in the polymerase. That's a really, really important first principle for antibiotic resistance, for drug resistance in general. Whatever the target protein is, if the gene mutates, the site where the drug binds, right, it's going to change the effectiveness of the drug. So that's a big, important first principle that you've just met. Okay. So we already said this. There are multiple types of RNA, most of which you've met previously in other undergraduate training. Okay. So let's focus specifically on the messenger RNA. How does the bacteria make messenger RNA? We're going to go into a little bit of detail here. And part of the reason to explain this again is keep this in mind later when we talk about mammalian transcription, because you'll see that this is pretty dead simple compared to mammals. So genes in both bacteria and mammals have regions upstream of the coding sequence called promoters. So a promoter is a piece of DNA to which the machinery that's going to start the RNA, the messenger RNA synthesis, will bind. That's how it knows where to go. Okay? And what's shown here is there's two little red spots. And these two little red spots are some distance from each other. And that's true in many bacterial genes. You'll have a spot that's about 10 nucleotides upstream from the start site. And in the old literature, it was called the Pribnow box. And then you'll have a second site, maybe 30 or so nucleotides away. And this second site is important because it's where this little character, this little protein called a sigma factor. This is not a single protein, it's a family of proteins that regulate transcription. So remember that, it's a family of proteins. And what the sigma factor does is it's sort of riding along with the RNA polymerase, which the best model is that the RNA polymerase is kind of scanning the genomic DNA, looking for something to do. And so when it comes across the Pribnow box and the other feature of the promoter, you can see the sigma factor binds. This then triggers the RNA polymerase to change shape. It clamps on and binds to the DNA. It begins to unwind the DNA, okay, in the process called initiation. Now, a couple things can happen. It can unwind the DNA, and for kinetic reasons or lack of fit or a variety of reasons, it can stop and simply abort and go back to the beginning. But if it's successful, what happens then is the sigma factor lets go of its other binding site, eventually dissociates, and the DNA template is then fed through the polymerase, and here you can see the RNA coming out at the end. So that's the elongation phase. Now, all things must come to an end. Good things come to an end at some point, and what happens is as the polymerase then finds the proper place to stop, you have the process of termination. In bacterial molecular biology, there's a row-dependent and a row-independent termination process. Row is a protein. It's actually a complex of proteins that bind to this complex when it's time for transcription to stop. Okay? The other way this can stop is that the polymerase can run into an AT-rich region of the gene, and what happens then is, because remember ATs are energetically less favorable than GCs, the whole complex can come apart. Okay? The RNA synthesis will stop, and there's a stem loop structure that's probably involved that causes this to all fall apart and to stop. So you have a protein-mediated stop signal, but you also have a DNA sequence-mediated stop signal as well. And we won't really worry about the details of when this happens and why and how. When you go to micro and you learn about specific organisms and specific disease processes, you'll learn a little more about the details of some of this. Right now, I just want to kind of give you a big picture. All right. The upshot then is this whole process is inhibited by rifampin because it binds to the one of the subunits of the polymerase. The rifampin binds to the polymerase, one of the subunits of the polymerase. All right, so this is a cool picture. I don't think it has any particular medical relevance, but it's pretty cool. So here's what's going on here. So there's a DNA, double-stranded DNA, right, going the length of this image. And what you see here, these are messenger RNA molecules coming out of the DNA. And these little black dots riding along here, this is the polymerase complex. Okay? So this is a whole army of polymerases essentially marching down the DNA, reading the sequence and generating RNA. Now notice out here at the ends of these RNA molecules, what do you got? You got these little black dots, right? So what's that all about? Well, this is to remind me to tell you that RNAs have a fair amount of secondary structure. Primary structure means the sequence, right? Whether it's A, G, C, U, G, A, whatever. The secondary structure means that anytime there's regions of complementarity, the molecule can actually fold itself up and form stem loops, in other words, stem and a loop. And in addition, it can form, sometimes between these loops, there's also hydrogen bonding that occur that produce a significant amount of secondary structure. So often in the cartoons, we think of messenger RNA as this nice little line and then, ribosomes chugging along it. Anyhow, it is. It's actually almost always bound with proteins, rolled up, folded up, and has significant secondary and sometimes even tertiary structure. Okay, so overview. What does transcription require? Promoter, which we just showed you. No primers, right? We talked about DNA polymerase, and I said, you got to have an RNA primer, it's got to have a 3' hydroxyl, or stuff doesn't happen in DNA synthesis. Okay? You can make DNA primers in experimental systems, and so forth, but you got to have a primer. RNA polymerase, don't need no stinking primer. You need GTP or ATP to start, and that's about it. It copies only one DNA strand, right? The template strand. The opposite DNA strand is the coding strand. The coding strand has the same sequence as the messenger RNA. You got that? Makes sense? Okay. So it copies one strand that's complementary to the template and identical to the coding strand of DNA. Now, just like a DNA polymerase proceeds in a 5' to 3' direction, but unlike DNA polymerases, important point here, unlike DNA polymerases, there is no 3', 5' exonuclease activity. So the error correction is not as good. There is some error correction. RNA polymerases can basically, so they'll catalyze a nucleotide, adding it to the RNA, and that reaction is actually reversible somewhat. Okay? But it doesn't have the exonuclease where it actually cuts it out and then replaces it with the proper base. So there's messenger RNA synthesis is much more error prone. This concept of RNA polymerases not having very good error correction has implications beyond just bacteria and messenger RNA. And this will come into play when you learn about virology and you learn about RNA viruses. RNA viruses, right, when the genome is synthesized, that tends to be very error prone. And so there's lots of mutations in certain types of RNA viruses, like HIV. Okay, very error prone, lots of genetic instability. And clinically, that has important implications because resistant strains arise rapidly within days, months, drug treatment. The possibility of making a vaccine against an organism with a very unstable genome is very low. Okay? So you see this fundamental principle that RNA polymerases tend to be error prone has implications for lots of different things. Okay. So that's worth understanding. All right. And I already made this point that RNAs can fold up into interesting structures. Some of them look like a little guy. All right. Okay. So the next point I want to make is to illustrate the general principle of how genes are organized in prokaryotes. And of course, it's the DNA sequence, the genes from which the messenger RNA is made. The structure in a prokaryote chromosome, shown here in red, basically, again, promoter, which we just talked about, and sometimes there's an additional sequence called an operator. That's going to be an important concept, an operator sequence. Sometimes the operator turns things on. Sometimes the operator turns things off. This particular example is this operator turns off the synthesis of proteins or expression of genes necessary for tryptophan synthesis. So it's called the trip operon. And this was the first repressor operon discovered in the 1950s. Now the point here is from this trip operon, one messenger RNA molecule is made. There are no introns, so it's just end-to-end, one long message. From that message then is translated a number of different proteins. Doesn't matter what they are right now, but what they do in this operon is they synthesize tryptophan. So the idea here is when there's a lot of tryptophan in the environment, the bacteria doesn't need to make the enzymes to generate tryptophan, right? Because it's being delivered from the grocery store. All right? When there's not enough tryptophan in the environment, bacteria can make its own if this gene is turned on. All right, so we'll talk more about operons when we talk about gene expression, but I wanted to introduce this idea to you of what is an operon. Now, the other type of gene that is expressed in bacteria would be genes that will code for RNAs themselves. Okay? There are genes that code for proteins, and there are genes that code for RNA. This is an example of the structure of E. coli genes coding for ribosomal RNA and transfer RNA. And again, you can see they're kind of it's this continuous sort of structure. Now, in this case, the blue box indicates the coding sequence for the 16S ribosomal RNA. And if you're not familiar with this nomenclature, 16S, S is a Svedberg unit. That's you spin stuff in a centrifuge, and its buoyancy and its density, you come up with a number. So the heavier than the denser, the bigger and the denser the thing is, the higher the Svedberg number. Okay? So in the early days, it was a way to isolate these particles. So that terminology is kind of archaic, but it's used even now. So anyway, this particular subtype of ribosomal RNA, coded by the blue box, is separated in space from this red coding sequence, which codes for transfer RNA of some sort, and this blue box coding for the 23S RNA. So these little yellow sections are sections that are not going to be transcribed. They're not necessarily transcribed. And so what happens is you get this initial transcript, these things get cut out, and then you end up with the mature RNAs, which then get assembled into the ribosomes. Okay? Now, the trick is, the important thing is in bacteria is often there are a whole bunch of copies of these genes, 6, 7, 8, 10, 12. And you think, well, why would that be? I mean, you have a protein coding gene, and you typically maybe have one, maybe you have two copies, maybe you have a homolog, but you don't have 6, 7, 8, 9, 10, not normally. Okay? Why is that? Well, the reason is that the product, the RNA itself, is directly used to form the ribosome. There's no translational amplification. You don't get extra copies. Okay? You just get what's made directly from the gene. So that's why there's a whole bunch of copies of the gene. Okay. And there's our buddy transfer RNA. All right. Now, the interesting thing about bacterial DNA, human DNA, viral DNA is transcription can actually occur from either strand. And it depends on the structure of the genome. Okay, so this cartoon is just to represent in E. coli an example of several different genes. Gene A, for example, is the message is transcribed in this direction. Gene B, it's transcribed in this direction. Okay, and so forth. We're going to learn even in mammalian systems that you can actually make RNAs in both directions from the same strand. And those RNAs have different products. And this is true in viruses as well. So the whole thing gets really kind of messy depending on exactly which molecules you're talking about. Okay, so let's kind of reinforce the notion that the regulation of the mRNA synthesis is medically relevant and important. And the story here is a story about how a bacterium will respond to environmental changes. Okay? And one of the classic sets of proteins that have been described for many, many years are called stress response proteins. And in the bacterial world, stress means a change in pH, a change in desiccation, a change in availability of nutrients, and so forth. It's probably a little bit of misleading to call them stress response proteins because they're not only responding to a stress, they often do, but they're just normal proteins that allow the bacterium to express different sets of genes depending on conditions. Okay, so they adjust to the environment. But that's their terminology. And they belong often to this family, this sigma factor family. And as I've already described, what they do is they interact with promoters. Okay? So not all genes get expressed all the time. So depending on which sigma factors are produced, in this example, this is one called sigma factor 32, and it will cause a certain set of genes to be expressed, a certain set of messenger RNAs to be produced. Now, here's an example of how this kind of has impact on clinical microbiology or clinical medicine. So this story is about a Pseudomonas infection in cystic fibrosis. So we really haven't talked much about cystic fibrosis, and we will in the future. But so cystic fibrosis is a lung disease, and it's a genetic disease. And it's a genetic defect, a mutation in a chloride channel. Okay? It's called the CFTR, chloride channel. You think, well, what's that got to do with lung disease? Because what happens in cystic fibrosis patients is they become very susceptible to infection. So what's the connection between a chloride channel, mutation of chloride channel, and infection? Well, the connection is water. So one of the things that cells do is they maintain sodium and chloride and potassium levels, right, by channels and ion pumps. And chloride is really critical for this whole process. And so when the CFTR channel is mutated, cells that normally maintain proper water excretion, like the cells lining the alveoli and the airways in the lung, that process becomes deranged. And what happens in the lung is that the mucus that's normally secreted in the lung becomes thicker and heavier because there's not enough water. Say, well, okay, what's mucus have to do with infections? The lung, one of its major jobs in helping the immune system is it's an initial point of defense against inhaled pathogens, right? You inhale dust, you inhale bacteria, you inhale viruses. On the way through the lungs, they stick to the mucus. It's kind of goopy. The ciliated epithelium kind of shove the mucus up into your mouth and you swallow it. And the acid in your stomach kills the bacteria. Right? That's a major first-line defense against infection. So what happens in CF patients? They don't have proper chloride channels. They don't secrete the right amount of sodium, potassium, and water. The mucus becomes thick. It starts to plug the airways. It can't be cleared. The bacteria and the stuff that are breathed in get stuck there, and they get infections. What kind of infections? All kinds of infections. One of the most serious and important ones is infection by a bacterium called Pseudomonas aeruginosa. Okay? So in this particular story, what the investigators in this old paper observed is that some of the Pseudomonas that cause the worst disease, they found out were upregulating a particular type of sigma factor called RPOH. So there's an increase in the stress response protein in this particular infection, which made it even worse, made it this mucoid phenotype, difficult to treat and very deadly. And so they think that the upregulation of this particular sigma factor contributed to inflammation in the chronic infection and the worsening of the prognosis of a patient with CF. Okay? So, that's kind of a very indirect story, starting from all the way back to Rho protein, or RPO protein, controlling messenger RNA synthesis in bacteria, but ending up with a patient with cystic fibrosis with a dangerous and difficult to treat Pseudomonas infection. Okay? So one of the things that we want to try to do in this opening module is to get you to start to think about those kinds of sequences of events. Not just to think about, what's a sigma factor? Oh, it's a protein that binds to a promoter in bacteria. Stop. Okay? It's not enough. You think, okay, if it binds to a promoter in bacteria, so what? Well, the so what is the bacteria now makes a different set of genes. So what? So the bacteria can become more dangerous for some reason, mucoid phenotype. So what? If the bacteria is more dangerous, then the prognosis for the CF patient is worse. Okay? Now, clearly, a physician treating that CF patient is not always going to drill all the way back to what sigma proteins do. Okay? It's kind of an interesting exercise, but it's not clinically useful. But going at this point forward, you need to be able to do that. You need to be able to do that to answer our test questions and to qualify in the board. So that's why we're doing some of these things. All right. So we're going to finish up. I just want to kind of foreshadow what we're going to do next. Is we've touched today on prokaryotes, pretty simple sort of process for transcription. But next time we're going to talk about eukaryotes, and there's a whole bunch more colored things here. Okay? Just to boil it down to what's important. We're going to have to know what the colored things are and how they work, and why this influences development, and why it influences disease, and why it influences therapeutics, treatment of disease, and so forth. So that's where we're going next. All right? So let's take a break. We'll reconvene at 10 o'clock over in TBL land. And at this point, are there any more questions about the TBL preparation? There were a couple at the break. Folks wanting to know about how much detail in the transcription coupled versus global repair, that kind of stuff. Big picture, you know. Recognize what nucleotide excision repair is about, how it works. Okay? What else? Anything else? Let me ask you at this point, a little feedback. Lectures are going okay, too fast, too slow, too much detail, too little detail. You want to know more about the biophysics of polymerases, right? They're actually pretty cool. When you think about the physics, when you move around with your muscles, right, you got this myosin generating all kinds of force. Polymerases make the same amount of force. It's ridiculous. But they're very cool. All right, anything else? You good to go? Okay, I'll meet you guys over there in a little while.